Hi everyone. So I'm a little bit sick, so I'm sorry about that. But my assignment is the smooth endoplasmic reticulum. Today I'm going to talk about basics. So it is found in both plant and animal cells. It's called a smooth ER because it doesn't have the ribosomes on its surface like the rough ER does. Um, its structure um, It has a network of tubules that connect throughout the cytoplasm. It connects the rough ER to the Golgi apparatus. Some cells are dilated like sacs in the rough ER, and it's more of a storage organelle. It has a lot of unsaturated fatty acids, making it very fluid. Its primary function is the manufacturing and metabolism of lipids, but all of its functions vary from cell to cell. It synthesizes phospholipids and cholesterol. There's a lot of detoxification processes that go with it. In the muscle cells, it can regulate calcium ion concentration, and it has a lot of steroid production and hormone synthesis. Um, it's also involved in the production of steroid hormones in the adrenal cortex and the endocrine glands. So where is it? Well, it's found in the cytoplasm, like the rough ER is. It's mostly found in the hepatocytes, which are in charge of detoxification and lipoprotein formation. It's also found in endocrine cells a lot, which is mostly in charge of steroid hormone production. Here are some pictures of it. So the top left is it shows the smooth ER, the rough ER, the nuclear envelope, and the nucleus. I included this picture because you can definitely see that the difference between the rough ER and the smooth ER, whereas the smooth ER doesn't have all those little dots, which are called ribosomes, on them, hence making it the smooth ER. Um, and the bottom right picture is out of a microscope, and so on the left is the smooth ER, and you can tell that they're a lot closer together, and you can see, much like in the top left picture, the little circles, like the little tubulars, and then whereas the rough ER down in the right bottom are much further spread apart and a lot rougher. I guess that makes sense, but yes. So as a little bit of comparison between them, the smooth ER is tubular, whereas the rough ER is obviously folded sacs. Um, they don't have ribosomes on them, whereas the rough ER has them on the membrane. Um, the smooth ER is mostly involved in lipid synthesis and calcium storage, whereas the rough ER is mostly involved in protein folding and maturation. Um, they're found in, like I talked about earlier in this other slide, I said they're found in hepatocytes and endocrine cells, where in rough ER, it's found in plasma cells and pancreatic cells. So, the smooth ER is actually around 150 nanometers in diameter on average. I mean, it obviously varies from cell to cell, they're not all the same size. But the rough ER is determined not in diameter, but in like deepness, and that's 100 nanometers deep. So there's that. Let's talk about research. So our teacher asked us to look at some research from like current things right now based on our topic. This is just one of the many websites I found. Most of the stuff about smooth ER has already been figured out, but right now they're studying the existence um, of domains between the ER and other organelles. They call this the MCS, or membrane contact sites, that they're trying to figure out how they react with each other. Um, they are focusing on contacts of the ER with the mitochondria and the endosomes, and I didn't find a lot more than that, but it was found from the Department of Molecular, Cellular, and Developmental Biology at the University of Colorado Boulder, which I thought was pretty neat considering, Colorado, right? And that's pretty much it for the smooth endoplasmic reticulum. So I hope you guys learned something. If you have any questions, please let me know. Thank you.